Hi everybody, I am Berlin Grace, working as professor in the Department of Biotechnology, Carnegie Institute of Technology and Sciences, Coimbatore. So today I am going to explain the overview about the cancer detection methods for the course Cancer Biology. So cancer detection methods, we have a series of methods. Here we have a patient who will be suspected to have cancer. Cancer is a disease which is not having any symptom at the initial stage. So how they suspect? So the symptoms, the basic symptoms start popping out once the cancer progresses. So based on that symptoms, or else some people will go for a regular screening. So when they go for a regular screening, they find the symptom of cancer. Or else for certain cancers like uterine cancer, people will be having other complications like overbleeding, etc. They go for hysterectomy. When they go for hysterectomy, by chance they find that they have cancer in the uterus. So by all these means, they have the suspicion that they may have cancer. So they approach a doctor. When they approach a doctor, the doctor also immediately cannot confirm that it is cancer. So they go for a physical examination. When they do the physical examination, so they can find some lumps or some extra growth in particular region. With that also they cannot confirm that it is the cancer. So it may be a benign tumor. So in order to rule out all this and to confirm the cancer, they have to go for further blood test and the analysis in the cancer site itself. So for that they refer the cancer patients to blood test laboratories. What they do in the blood test? In the blood, when we are having a cancer, certain markers will be circulating in the blood. So that is a very easy method to detect. Instead of pricking the cancer site, instead of the tissue from the cancer site, which is more tedious process, we can test these markers in the blood. So which are these markers? These markers are nothing but the abnormal or overexpressed or changed molecules which are circulating in the blood. For example, they are simply called as antigens. What is antigen? Antigen is a molecule which is able to produce antibodies against using the help of immune system. So these antigens are expressed abnormally, so considered as a foreign bodies. Certain tumor markers are circulating in the blood that we categorize into two categories. One is tumor specific antigen and another one is tumor associated antigen. So what is tumor specific antigen? Tumor specific antigens are expressed only in the tumor tissue. It is not there in the normal condition. So in the tumor tissue it is produced and it is circulating in the blood. The other category is tumor associated antigens. That can be the normal molecule which is present in the normal condition as well as in the cancer condition. So what is the difference? In cancer condition the level will be high. So that is also a tumor associated antigen. So tumor specific antigen, we have lot of tumor specific antigens like if an virus infect, human papilloma virus infect particular patient, so that viral proteins will be the antigen. So that is expressed, and similarly tumor associated antigens, we have certain enzymes. In skin cancer it is due to the excess melanin production. Melanin synthesizing enzyme is tyrosinase. So when the tyrosinase is high level of melanin production will be there, that is a marker. So similarly for tumor specific antigen we have mutated proteins. Normal protein which are controlling the cell growth are called tumor suppressors. These normal proteins, when it is mutated, that will not do the function. So these are called as tumor specific antigens. So likewise, so many tumor specific antigens are there and so many tumor associated antigens are there. Particularly very commonly carcinoembryonic antigens, alpha fetoproteins, these are the commonly used markers which are identified in the cancer patients. And we have a specific marker for prostate cancer called PSA, prostate specific antigen. And these antigens are tested using several techniques. One such technique is ELISA, enzyme linked immunosorbent assay. So where the cancer patient's sample will be having the particular antigen and we have an antibody specific to that. These combinations in a 96 well plate, when we put it, the reaction product will be detected with the help of color formed, brown color formed, and that color will be determined using calorimeter at particular nanometer, and that gives the level of the particular tumor marker. And then we have another technique called radioimmunoassay, where we have already a known level of antigen and known level of antibody which are labeled, radio labeled. So this will give radioactivity. Higher level of radioactivity can be observed at the initial stage, and then when you add the sample, this will replace the radio labeled antigen. The antigen present in the sample will replace the radio labeled antigen. So what happens? The radioactivity decreases. So from this we can find out how much antigen is there in the sample. That is nothing but tumor marker. So that is one method. And another method, detection using the biosensor strips. You may have seen certain strips for detection, like pregnancy testing strips are there. Similar to that, we can have that antigen antibody reaction. Based on that, if the positive marker is present, that will give the positive strands. So using the strips also you can detect the cancer, or else simple assay. If it is an enzyme like tyrosinase, we can go for its no substrate reaction method is there. Using calorimeter we can detect it. That is simple assay for the enzyme, if the tumor marker is an enzyme. And several other enzymes are also acting as the marker, like telomerase. Telomerase enzyme is an enzyme which is switched off in the normal autosomal cells, but during the cancer it is activated. And similarly many other tumor markers are there, tumor specific markers. So these markers are expressed in the embryonic stage, but in adult stage it will not be expressed. So here during cancer these embryonic antigens will be expressed. So inappropriate expression of particular protein or antigen is also a tumor marker. We have carcinoembryonic antigen, alpha fetoprotein, all these are coming under this category. So once the blood test tells that that patient is having some tumor markers, that alone is not sufficient, because lot of limitations are there for these tumor markers too. So these tumor markers are not very specific for particular cancer. One carcinoembryonic antigen may be expressed for 2-3 different cancers, as well as this also can be expressed if you have any liver damage or lung damage. So it is not very specific for the cancer alone. So we have to go for further techniques. And then physical examination can examine the cancer which is present in the visible regions like oral or skin or any visible regions. We can detect it. But if the cancer is in the internal organs like lung or liver, then you may have to go for further imaging techniques. It may be in the stomach where you cannot visualize it. So for all this we have to take an image of those cancers using certain aids. So how can we take that image? Imaging techniques. We have lot of imaging techniques. These are again categorized into non-radiation imaging techniques and radiation imaging techniques and nuclear imaging techniques, and many other immunofluorescent methods are also coming up. So how can we take that image using non-radiation methods? The most commonly used are ultrasound sonograph, where using the sound waves we can find the difference in the mass or cancer mass present in our body. And then we have an endoscopy, where if it is the cancer is in the stomach, we can insert a catheter like tube, and through which we can have an imaging material and which can take the images of the stomach, and then it will give the cancer whether it is present or not. And then we can see the radiation methods where the normal X-ray that also will give the cancer detection, and then MRI scan and CT scan, all these are radiation methods. And then further we have nuclear method where we have the positron emission tomograph which can give the scan called the PET scan. So these imaging techniques also will give some kind of information whether the cancer is there, where it is, and the almost the size. Not exactly, to some extent it gives the size of the cancer. So then further we have to confirm whether that cancer has spread nearby or in the faraway place. So for that we have to test the lymph nodes. The spread is indicated by the number of lymph nodes involved in the cancer. So that can be tested by taking biopsy from the lymph nodes and spread it in the slide like this. Biopsy can be spread in the slide, and this can be stained with hematoxylin eosin to find out how many cancer cells are spread to this lymph node region. And similarly we can take several lymph nodes nearby the cancer tissue and count how many lymph nodes are involved in the cancer. So depending on that we can find out the size using that cancer stage. They will give the cancer stage based on the number of lymph nodes involved and size of the tumor, and then whether it is metastasized or all that can be determined using this lymph node testing. And then they suggest the cancer patient to go for biopsy testing. So that is almost a confirmation, that gives almost a confirmation. Exact stage and exact type of cancer also can be determined after you analyze using the biopsy. So biopsy is nothing but taking the sample from the cancer site itself. That is little painful process only, but since all these remaining imaging techniques and blood tests are confirming that that person is having cancer, they have to go for biopsy. So biopsy, if it is taken from the liver or any soft tissues, that they will take only in the fluid manner. They take a little fluid using needle, that is called a needle biopsy, and that also again they spread it in the slide and they can stain it with the hematoxylin and eosin to find out whether abnormal cells or cancer cells are there and how many cancer cells are there. All that can be analyzed by a trained histopathologist. And then if it is a solid cancer like uterine cancer or any stomach cancer or anything, then they have to take a tissue. So using a cone, that is called a cone biopsy, a piece of tissue can be taken from the cancer tissue, and that we need to put it in the 10% formalin, buffered formalin. It is not simply formalin. If you put it in the formalin, it will destroy the cells completely. So we cannot study that, we cannot analyze it later. So it is better to put it in 10% buffered formalin so that it will preserve the cell structure and cell organelles, etc. And fix it in this way, then that tissue like this, it may be preserved in the buffered formalin within 10 days to 1 month. We have to then block this tissue, otherwise it will be destroyed completely. We have to block it in the paraffin baths. The tissue can be then taken and blocked in another mold which is having the wax in it. You just melt the wax, pour it, and immediately you place the tissue inside. So when you place the tissue inside and once you make it solidify, that tissue will be seen like this. So once this blocks, once this paraffin wax is solidified, after a day you can take that blocks outside also like this. You have the tissue and the paraffin wax is there, and you can even give the label at one side with some number or some that cancer patient's code, something like that you can give, and the details can be written in another paper for your reference. It can be written in another paper and kept for your reference. So then this can be kept for even several years. Whenever you want to take the section to study, you can take it. How do we take the sections? We have to place this in a microtome. You have a microtome in the laboratory. So this will be having an apparatus where you can keep this mold. You can keep this mold, exposing the tissue outside, and you'll be having a blade. When you rotate it, this tissue will come, it will move up and down so that it will be cut, and then that tissue you can put it in the hot water. So immediately insert one slide and you can take it in that slide. So then this slide you can then keep it in the oven to dry it, and this slide will be having the section along with the paraffin. So now you have to remove the paraffin. In order to remove the paraffin, then you have to keep it in the oven, in the incubator, and treat it with the xylene to remove the paraffin, and then further it is already dehydrated tissue. Now we have to rehydrate it to stain this with hematoxylin used with a series of alcohol reagent, and then finally with water and xylene you have to wash the tissue section so that it will be completely dewaxed, and you can add the hematoxylin. The hematoxylin will stain the nucleus and the eosin will stain the outside of the cell, which means the proteins and all that will be stained with the eosin. So that gives the contrast between the cells and the extracellular matrix, membrane, all that. So you can view this slide under the microscope and see how many cells are quite normal and how many cells are cancerous. So you can count the cells, number of cells, and the massive growth of the cancer tissue can be seen under the microscope. With that we can detect the cancer. And further, the same tissue can be even used to detect the tumor markers. I told you that certain tumor associated markers and tumor specific markers, antigens, are present in the cancer tissue. That can be detected with the blood test. In the cancer tissue also similarly we have so many specific markers, molecular markers. Those molecular markers can be determined using the specific antibody that can be stained in that section. That will give the brown colored section. After observing under the microscope, we can count the positive cells for particular antigen. So that gives whether that antigen is associated with the cancer. For example, breast cancer itself, if you take, it is having different subtypes. In that subtypes, it is all based on the molecular markers. So depending on the molecular markers, we can decide the treatment method using different molecules. Molecular therapy can be done based on this molecular detection. This molecular detection I will explain in the next class in detail. Hope you all understood. Bye.